Chimeric antigen receptors are synthetic modular proteins that redirect immune cell reactivity toward a target of interest. 6 CAR T cell products have been approved by the U.S. Food and Drug Administration for 12 indications. However, various barriers restrict the efficacy and/or prevent the widespread use of CAR T cell therapies in these patients as well as in those with other diseases, particularly solid tumors. The evolution of CAR designs beyond conventional structures will be necessary to address these limitations and expand the use of CAR T cells to a wider range of diseases. In this presentation, we discuss the progress in the development of innovative designs for new CAR T cell products to increase and expand the clinical benefits of these treatments for patients with various cancers, and we explore the potential of CAR T cell therapies in addressing diseases beyond cancer. We will discuss the following topics, including basics, key milestones and challenges in CAR T cell therapy development and next-generation CAR enhancements and their corresponding platforms designed to address the challenges, as well as the potential of CAR T cell therapies in addressing diseases beyond cancer and the clinical status. Prototype CARs comprise a target binding domain, such as a scFv, a hinge or spacer domain that projects the binder away from the cell surface and provides conformational flexibility, a transmembrane domain that anchors the receptor on the cell surface, and costimulatory and CD3 zeta signaling domains that provide activation signals. Small modifications in CAR structure can have profound effects on CAR T cell function, including modulation of the antigen density threshold for activation, persistence, potency, tonic signaling, CAR expression level, and the propensity for dimerization. At present, autologous CAR T cell manufacturing requires extensive infrastructure, time, and lymphodepletion of the patient. Allogeneic CAR T cell manufacturing can potentially reduce the associated costs and time required, but still requires lymphodepletion of the patient. In vivo platforms, such as targeted viral delivery and targeted lipid nanoparticle delivery, are much less mature, but have tremendous potential to disrupt this industry by potentially obviating the need for manufacturing facilities and lymphodepletion. Targeted lipid nanoparticle delivery has the additional feature of only transiently expressing the CAR. CARs were invented by Eshar and colleagues with the goal of harnessing the expansion, killing, and persistence of natural T cells while overcoming major histocompatibility complex restriction of the T cell receptor to enable broader therapeutic applicability. Since the first-generation prototype of chimeric antigen receptor was described in 1993, CAR T cell therapy has transitioned through the concept phase, prototype iteration, clinical proof of concept, and has successfully commercialized in the clinic for the treatment of certain subsets of hematological malignancies. Although treatment with CAR T cells has produced remarkable clinical responses, many challenges limit the therapeutic efficacy of CAR T cells in solid tumors, hematological malignancies, and other diseases beyond cancers. Although initially single antigen targeting CAR T cells deliver high response rates, a significant portion of malignant cells in treated patients display partial or complete loss of target antigen expression, known as antigen escape. In addition, in the case of treating acute lymphoblastic leukemia/lymphoma with CAR T cell therapy, toxicities have been observed in nearly all patients. For solid tumors, as solid tumor antigens are also expressed on normal tissues, antigen selection is crucial in CAR design to ensure therapeutic efficacy and limit on-target off-tumor effects. In the tumor microenvironment, many immunosuppression driving cells can infiltrate solid tumors, including myeloid-derived suppressor cells and regulatory T cells. These infiltrate tumor cells and drive the production of tumor-facilitating cytokines, chemokines, and growth factors. In addition, immune checkpoint pathways such as PD-1 can serve to decrease anti-tumor immunity. Compared with hematological malignancies, CAR T cell therapy for solid tumors is hindered by the limited ability of CAR T cells to traffic to and infiltrate solid tumors as the immunosuppressive tumor microenvironment and physical tumor barriers block the way. Investigators are addressing the current obstacles of CAR T cell therapy with a wide range of engineering strategies to improve the safety, efficacy, and applicability of this therapeutic modality. Reported strategies for next-generation CAR enhancements include multispecificity and/or logic gating, fitness enhancements, regulatable platforms, armoring, engaging the endogenous immune system, expansion and persistence, alternative signaling, stealth or fratricide-resistant, and alternative immune cells. In addition, distributed manufacturing and allogeneic products, and next-generation gene delivery such as CRISPR-based gene delivery, transposon-based gene delivery, and nanoparticle-based in vivo gene delivery, are also considered to enhance the access and efficacy of CAR T-cell therapy. Based on the next-generation CAR enhancement strategies, several novel innovative platforms have been established to accelerate the development of next-generation CAR T-cell therapies. Investigators have leveraged numerous bioengineering strategies to develop advanced CAR platforms to improve the safety and efficacy of immune cell therapies. Multispecific CARs, which operate as an OR gate, may be able to overcome obstacles related to tumor heterogeneity and antigen loss, whereas combinatorial antigen sensing systems such as SynNotch or LinkCAR increase the specificity of CAR T-cells by requiring two antigens for activity. Safety switches such as drug-regulated and adapter CARs could mitigate A-induced toxicities and enhance efficacy by tuning signaling. Knockout of TRAC, B2M and 2 genes ablates TCR, MHC class I and MHC class II expression, respectively, and HLA-E and CD47 overexpression shield stealth CAR T-cells from natural killer cell and macrophage-mediated rejection. Deletion of NR4A and TET2 genes or ectopic overexpression of c-Jun results in transcriptional rewiring that renders T cells exhaustion resistant. By fusing an scFv domain to the TCR subunit, TRuC and HIT receptors redirect the specificity of the endogenous receptor and exhibit increased antigen sensitivity compared with CARs, while secreting lower levels of cytokines. Integration of a cytokine gene in the CAR vector provides a growth and survival signal to improve the persistence of T cells. Although most of the novel CAR designs are under the proof-of-concept status, several novel CAR-T platforms are in ongoing clinical trials. Armored CAR is produced by co-expression of various proteins and ligands, or co-secretion of cytokines with the CAR to enhance CAR T-cell function. Logic-gated CAR is based on Boolean logic-based signaling to respond to multiple inputs to alter signaling or expression of CAR and/or proteins. This includes OR, NOT, and gates, but not dual targeting, which is categorized under multi-antigen targeting. While multi-antigen targeting CAR is designed for simultaneous engagement of multiple antigens for increased specificity. On-off switch CAR can turn engagement with target cells on or off with various mechanisms. And switchable CAR can engage multiple antigens using a single CAR-T with adapter moieties. These innovative platforms, together with conventional CAR designs, show potential to treat blood cancers, solid tumors, and autoimmune diseases via autologous or allogeneic transfer. The clinical success of CAR T therapy has incited the imagination of those outside the field of cancer. Theoretically, CAR T cells will find utility in contexts in which eliminating a particular population of cells is therapeutic, and such a population of cells can be targeted specifically. These two premises, although simple, are a limiting hurdle for many diseases. B-cell and plasma cell malignancies reveal that such an approach is possible, but identifying a targetable antigen for other cancers has proved difficult. Cancer presents several substantial barriers to CAR T-cell efficacy, including a large target burden, nearly total clearance of malignant cells, a high mutational load, and a hostile tumor microenvironment. By contrast, other diseases generally have a much smaller target burden, partial clearance of diseased cells can be therapeutic, a lower mutational load, and are generally not localized in such an inhospitable environment. Emerging clinical and preclinical evidence suggests that CAR T cells may be used to treat autoimmune disease, chronic infections, heart failure, and other chronic conditions. Currently, only 4 CD19-targeted CAR-T therapies and 2 BCMA-targeted CAR-T therapies have been approved by the U.S. Food and Drug Administration for the treatment of certain types of hematological malignancies. Axicabtagene ciloleucel was the first FDA-approved CAR-T therapy. It targets CD19, a biomarker for normal and neoplastic B cells, and is critically involved in establishing intrinsic B cell signaling thresholds through modulating both B cell receptor-dependent and independent signaling. CD19-targeted CAR-T therapies have been approved for the treatment of specific patients with large B-cell lymphoma, follicular lymphoma, mantle cell lymphoma, and B-cell acute lymphoblastic leukemia. BCMA is a member of the tumor necrosis factor receptor superfamily that plays an important role in regulating B cell proliferation and survival. BCMA-targeted CAR-T therapies have been approved for the treatment of multiple myeloma. In addition to CD19 and BCMA, there are also numerous targets of CAR-T cell therapies with clinical evidence of efficacy. CAR-T cell therapies that target CD20, CD30, CD38, CD7, and kappa light chain have been reported to be promising in ongoing clinical trials for the treatment of other hematological malignancies like Hodgkin lymphoma, non-Hodgkin lymphoma, acute myeloid leukemia, chronic lymphocytic leukemia, and T-cell acute lymphoblastic lymphoma and leukemia. GD2, HER2, EGFR, PSMA, mesothelin, Claudin 18.2, and IL-13R alpha-2 are hot targets of CAR-T cell therapies designed for the treatment of solid tumors like neuroblastoma, sarcoma, glioblastoma, diffuse midline glioma, malignant pleural disease, gastric cancer, pancreatic cancer, biliary tract cancer, and metastatic castration-resistant prostate cancer. Notably, CD19-targeted CAR-T cell therapy, which was originally designed to treat hematological malignancies, has been reported to be effective in treating systemic lupus erythematosus. Creative Biolabs is a world-renowned service provider for immunotherapy. We have one of the world's largest collections of CAR products of different generations targeting various biomarkers, and we continue to innovate the next generation CAR technologies to achieve even greater results. Based on advanced technology and years of research, Creative Biolabs offers high-quality custom services covering the entire CAR-T therapy development process to best suit your technical, program and budget requirements, greatly assisting your research, preclinical investigation, and clinical stage development. Our end-to-end CAR-T therapy development services include, but are not limited to, biomarker identification and selection, high-affinity scFv generation, CAR design and construction, virus packaging and CAR gene delivery, CAR-T in vitro assay, and CAR-T preclinical in vivo assay. As a trusted partner in the discovery and development of CAR-T cell immunotherapy, Creative Biolabs provides collaborative approaches and various solutions to help our clients bring a novel therapy to the clinical phase. We are committed to providing custom in vitro and in vivo studies and solutions to reduce potential risks and accelerate your CAR-T therapy development projects. If you are interested in learning more about Creative Biolabs cell therapy-related products or services, please feel free to call our service hotline or visit our website for more details.